So, hello and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koster, and we'll be talking about the discovery of small molecule HPK1 inhibitors for immuno-oncology with Rebecca Gallego today. Good morning, Rebecca. How are you?
I'm good. Thanks for having me.
Awesome. Yeah, thanks for coming. And now, without further ado, I want to introduce our guest, Rebecca. So, Rebecca Gallego is an associate research fellow within the oncology medicinal chemistry team at the Pfizer R&D site in San Diego. Her work focuses mainly on small molecule drug discovery, with particular interest in design strategy and the use of quantitative efficiency metrics to inform drug design. She received her PhD from the University of California in Berkeley in the lab of Richmond Sarpong, where she was supported by the ACS Division of Organic Chemistry Graduate Fellowship. Rebecca also serves on the board of the Medicinal and Bioorganic Chemistry Foundation, MBCF, which organizes the famous Winter Conference on Medicinal and Bioorganic Chemistry, for example, in Steamboat, happening next January. It's an honor to have you here, Rebecca, today. So welcome again.
Thank you so much.
Awesome. And in the good old Drug Hunter tradition, we love to get to know our speakers a little bit more. So we'll ask some warm-up questions here. So Rebecca, what inspired you to start a career in drug discovery?
So the big picture was I liked solving problems and I like helping people. So I feel like, going into the pharmaceutical industry, drug discovery is a natural fit with that. But the specific catalyst was taking an organic chemistry course in my freshman year at the University of Rochester. And it was a smaller course available to students who had taken, certain chemistry courses in high school. And it kind of just clicked with me, which I think a lot of people on the call will relate with that feeling. And ultimately, it inspired me to join a laboratory there. I was in Robert Beckman's lab undergraduate, and I wanted to go on to graduate school. And as you said, I was in Richmond's lab at Berkeley, and kind of was focused on that big picture goal of finding a way to solve problems and help people through drug discovery from that initial course I took.
Amazing. Yes, that shows us how important education is today. I love
Mhm.
that. Love that. What's your favorite molecule, Rebecca, and why?
Yeah, so it's hard. I think we all have a few favorite molecules, the ones that we work the hardest for, probably. So, um, the one that I'll speak about today is absolutely one of my favorites. It's a compound that I helped contribute to as part of a big team, and it advanced to the clinic, as an attempt to try to help patients, which is something I find a lot of meaning in. The other, favorite compound I have is megelanine, the natural product I worked towards in graduate school. And I never made it, but you put in a few years trying to figure out all sorts of ways to put it together, and you know, nature does it so beautifully and so simply, and we try with organic chemistry to create that. And although I never made it, it just holds a special spot in my memory, I'd say.
Amazing. Amazing. Yeah, that's wonderful. Yeah, we're looking forward to hear, of course, much more about the HPK compound and how it was discovered. I think that shows the problem-solving skills that you've developed throughout your career. Yeah, super, super impressive campaign. What are you most excited about in drug discovery today, Rebecca?
Yeah, I mean, we start off often as organic chemists and then move into medicinal chemistry. And what's really exciting about medicinal chemistry is it feels like both an established and an evolving field. So there's a lot that we can learn and do learn as we start along our career paths. And then I feel like there's a lot to kind of discover and share. So that could be, case studies and all that we share in the literature and in talks. And it's also kind of the fundamentals of the strategy of how we pursue medicinal chemistry. We all have these rules, rule of five, and now we're going beyond rule of five. Our modalities are changing. And so we're kind of creating that next level foundation for the future practitioners of our field. So it's a kind of cool place to be on the interface of like a very established field and a changing field.
Yeah, no, that's super important. Again, highlighting the knowledge transfer and sharing what we've learned and applying this towards the common goal of the industry to discover life-changing medicines. I love that. Yeah. And if you could go back in time to kind of the beginning of your career and give your younger self some career advice, what would you say to your younger self and maybe to some of the younger folks in the audience here starting out their career in drug discovery?
Yeah, I mean, we all focus on honing those technical skills. There's so much to learn in our field, and a lot of science is that technical part, but there's huge value in the soft skills and how you learn, to communicate science, that type of thing. And um, that's not just within our field of other, organic and medicinal chemists. As you evolve into drug discovery, it's across different fields. You're speaking with biochemistry and biophysics, biology, DSRD, PDM, you have to learn to communicate across all of these fields. So the more you can early in your career work on communication, and I think sharing your work in these broad settings, the better. And similarly, learning to work in a collaborative environment. So seeking out those projects where you're working with other students and postdocs, both within your lab and when available, like across labs. I think those investing in those things early are a huge benefit in terms of how you translate them to these like bigger team settings in drug discovery.
Awesome. Yeah, that makes a lot of sense for sure. I think I had to learn that as well.
We all do, yeah, for sure.
So, last but not least, what do you most like about Drug Hunter?
Yeah, I mean, you probably heard me hit on it a few times already today. Communication of science is hugely important to me. It's something that I like to invest in, either presenting or sharing, publications in the literature. And I think Drug Hunter is very good at that. There's kind of a characteristic trademark of making complex science both accessible to students and interesting to more, advanced scientists, more advanced in their career. So that's a hard balance, and I think the more that we can do that as a community, the better.
Awesome. Yeah, greatly appreciate that, of course. Awesome. Yeah, I want to, before we get started with Rebecca's presentation, I want to very briefly show folks one little trick here of how they can use Drug Hunter, and that's the structure search. So oftentimes when we're looking for like FDA approved drugs, that means that we have to browse several different websites to find out what kind of, structures are actually, what kind of motives are in FDA approved drugs. And yeah, with the Drug Hunter structure search, you can actually very quickly find out. So if you wanted to know FDA approved drugs that contain, let's say, pyridine, and maybe you can also use multiple functional groups. So pyridine and cyclopropyl groups, you can just search that very quickly here, and you will find them indexed here with the structure, the target, the year of approval. And you can actually quickly click them to get to the FDA website and immediately start more research here on the FDA's page, looking at, for example, the label and other things as well. You can also look at modern clinical candidates. So if you, for example, have the question, you want to look at phosphine oxides, but not phosphates, you can actually edit the hydrogen bond count here as well. Let's set that to 0 for the phosphorus to make sure we're only looking at phosphine oxides. Then we can check the modern clinical compounds here, and we can search. And we can, again, find compounds here. And what I find fascinating about those phosphine oxides is they're one of the strongest hydrogen bond acceptors here. So they often are flanked by an intramolecular hydrogen bond donor to make sure that those compounds are still permeable. So that's a really nice kind of design strategy. If you actually click here, you'll be guided to the PubMed page where you can learn more about any assays that this compound was run in or any bioactivities as well. And then last but not least, you can also do of course, a search on the Drug Hunter platform. So for example, if you wanted to learn more about drugs that contain acetylenes and cyclopropanes, which are two uncommon functional groups in drugs, of course, you can very quickly go here and learn more about those modern drugs that contain those. And if you click here, you'll be directly guided to a case study that maybe explains actually why those specific groups were used in this case. And now, without further ado, I will hand it over to Rebecca to share her presentation on the discovery of a small molecule HPK inhibitor for immuno-oncology.
Thank you so much, Dennis. Let me just pull up our slide. Great. Well, thank you all to everyone for signing on today. And thank you to Drug Hunter for the opportunity to participate in the Flash Talk series. I'm really excited to be a part of the scientific lineup that's participating in these Flash Talks. So today I'll be speaking about the discovery of a small molecule inhibitor of HPK1. And I'm not only going to discuss the lead optimization to identify the clinical candidate, but I'm also going to be speaking about the medicinal chemistry strategy and how specific metrics can enable multi-parameter and ultimately dose optimization. So I'm going to begin with the concept of lipophilicity, of course, how greasy or polar your compound is. Lipophilicity is of fundamental importance because it impacts many of the parameters relevant to drug design, those we optimize, but it impacts them often in oppositional directions as related to dose. So this concept has been published on by Merck, and they have a really nice JMEG Chem article that came out in 2020. So their graphical abstract is shown here. And to illustrate that oppositional impact on dose, for example, increased lipophilicity often leads to increased permeability, which can increase your bioavailability. But that same increase in lipophilicity often decreases your solubility, eroding your bioavailability. And this kind of oppositional impact is present across multiple, parameters that go into dose. So some of these parameters we optimize, like solubility, could be considered non-specific in nature, whereas others, such as potency, are specific. And by that, I mean reliant on a unique chiral environment. So to spend a little bit more time on this concept, I've illustrated another non-specific environment, which in fact is just how we measure shake flash log D. We put our compound in a bilayer of a water and organic phase, shake it up, and as it separates out, the log of the difference in concentrations in those two environments is your log D. So fundamentally, how it partitions. Then on the right, I've shown a specific environment being a protein, again, with those unique chiral amino acids. And this specific environment is actually very similar to the shake flash log D assay in that a binding pocket is a relatively hydrophobic environment by virtue of the greasy residues that are there within water. So there's kind of a commonality between these two systems, and that is that the relative lipophilicity will drive the partitioning or binding event. And so this is referred to as the hydrophobic effect. And it's this hydrophobic effect that fundamentally drives binding. And recognition of this is what goes into the concept of lipophilic efficiency. So before I move on to that, I want to describe another kind of fundamental importance around lipophilicity, and that is that it not only impacts all of these parameters that impact dose, but it also correlates strongly with promiscuity and odds of toxicity. So kind of a seminal medicinal chemistry paper from Hughes and Blagg et al. Came out in 2008, and they derived the 75-3 rule. And so they showed that the compounds that were most lipophilic with the least redeeming features, least redeeming polar features, had the highest odds of promiscuity. So that's the bottom right part of this table. And those are compounds with C log P greater than 3 and TPSA less than 75, hence the 75-3 rule. So this concept has been, noted in the literature across many publications. I have a second observation of that trend here. And in this 2013 MedChem communication, they show that with increasing C log P, there's increasing promiscuity. So that's the blue bars, where the red squares indicate the number of compounds in that space. So that kind of leads to a conclusion that if you consider toxicity profile and dose as very important parameters for drug discovery, getting the most potency possible for lipophilicity could be advantageous. So that's where LipE and LipMedi come in. Lipophilic efficiency is a metric that normalizes potency by lipophilicity to enable understanding where you're getting the most potency per unit lipophilicity. So I've shown a LipE plot here for those who may be unfamiliar with them. We put log D on the x-axis and negative log of potency on the y-axis. So this is a cellular readout that I'll refer to later in the talk. The P here is indicating negative log. LipE lines with a slope of 1 extend along the diagonal. And so compounds that cluster along those lines have the same lipophilic efficiency. One may be more potent, but it potentially might be because it is also more lipophilic. Utilization of this paradigm involves identification of SAR trends that lead toward the upper left-hand portion of this plot so that you can use them strategically as you pursue your other multi-parameter optimization goals. So LipE should be used in parallel with LipMedi or lipophilic metabolism efficiency. And ultimately, it does the same sort of normalization by lipophilicity, but for clearance. So you can kind of understand your profile in regards to log D changes. I will comment, as you tend to block soft spots with stable grease, you tend to improve your LipMedi. So we suggest, herein that use of LipE and LipMedi together are intuitive tools that can enable multi-parameter and dose optimization. So this sort of, use of these metrics has been stated in the literature across multiple publications. What we sought to do, however, was provide more of a quantitative assessment. And so towards that, we enabled a CDOS tool within Spotfire. So Spotfire is our data analysis tool. It's essentially a fancy Excel sheet where you can very readily put together plots such as the scatter plot on the left. And so CDOS is a column within that Spotfire. And what it does is it leverages dose equations. And so we're able to come up with a milligram per day endpoint based on measured and calculated inputs. So we typically, for any compound we're calculating CDOS, have measured potency as well as clearance. And we often use the hepatocyte assay for that. We're often calculating binding, blood to plasma ratio, and volume of distribution. The way in which I like to use CDOS is via assuming complete absorption, and that allows for the assessment of the interplay of potency and clearance, as has been suggested, can be done with LipE and LipMedi, but you're getting a milligram per day endpoint. So on the example on the left, I've shown negative log of cellular potency versus CDOS, and it's a milligram BID value on the y-axis. And this is for Cmin coverage over IC90 for this specific example, but you can tune it to CAV, Cmin, whatever threshold you'd like. So herein, we're using this column within Spotfire as a tool to assess the correlation of our efficiency metrics with dose. So the correlation I'm going to show you is CDOS relative to the sum of LipE and LipMedi. And we're doing that because, as I said, these metrics should be used in tandem. On the right is that correlation where I have LipE and LipMedi on the x-axis relative to CDOS. This is for a series of azolactam inhibitors of HPK1 that we'll be discussing today. And this was a CAV-based coverage paradigm. So as you can see, this is a remarkably tight correlation, and we believe provides proof of concept of the value of utilization of these metrics. That is, if you're systematically evaluating your SAR through a lens of LipE and LipMedi and recognizing SAR trends that increase those values, it will lead you toward a lower dose. So to change gears, I'm going to introduce HPK1. HPK1 is a kinase that acts as a negative regulator of immune cell signaling. So it puts the brakes on the immune system. And ultimately, our therapeutic hypothesis is that we can inhibit HPK1 and remove the brakes on the system to allow the immune system to exert control over the tumor. So in my mind, it's a very elegant solution to the need for novel oncology treatments, as it allows the immune system potentially to create the therapeutic effect. So to provide a little bit of context to how this might work, shown here is the cancer immunity cycle as published on by Chen and Melman. It begins within the tumor, where the cancer cell can release antigens, these peptidic particles. These antigens are then transported to the lymph node by antigen-presenting cells or dendritic cells, whereupon they can initiate priming of T cells. These activated cytotoxic killer T cells can then traffic to and infiltrate within the tumor, whereupon if they recognize that specific antigen that initiated the priming event, they're able to mediate cancer cell killing. So this is where some of the immunotherapeutic agents you may have heard of, Keytruda and others, anti-PD-1, anti-PD-L1 immune checkpoint blockade, large molecules work to extend existing anti-tumor immune response. The kind of gap with this is they only function when there's enough of a T cell signal to amplify. So all of these other steps have to be functioning well. This is where HPK1 comes in. So it's a kinase within the cytosol of the immune cells, such as T cells, and it puts the brakes on specifically when there's a weak signal. So on one hand, this could protect from autoimmunity. On the other hand, if there's a weak antigenic stimulation and the immune system is trying to recognize the cancer, HPK1 is going to shut that down. So this leads to our therapeutic hypothesis, as I mentioned, that inhibition of this kinase could lead to immune-mediated control of the tumor within cancer patients. So I'm going to start off with early lead that was derived from a virtual screening hit. Compound 1 is an amide that has fairly strong biochemical potency against HPK1 at a relatively low log D, which leads to a lipophilic efficiency of 7 in our biochemical assay, which is on the higher side. So this compound looks fairly appealing from that perspective, but we determined early on that this compound, as well as others in this series, suffer from human plasma instability. So that's, of course, a problem if our goal is an orally bioavailable inhibitor that has decent exposure to cover the target sufficiently. We were able to determine that we're likely undergoing amidase-mediated cleavage of this amide bond as the mechanism of instability. And what the team chose to shift to were these lactams as exemplified by compound 2. And you can see that they're stable in human plasma, which is great. We are, however, incurring about 100-fold loss in biochemical potency. If we also recognize that this compound has about 1 unit lower log D, we can take a look at the efficiency of this analog. And while it is less efficient, it's not as reduced in lipophilic efficiency as you might expect given 100-fold loss in potency. So we did recognize this as a viable starting point for continued optimization efforts. And so we moved forward with this lactam series as opposed to the plasma unstable amides with higher potency. So we were able to use structure-based drug design for this program and thus obtained a number of co-crystal structures, some of which I'll show throughout this talk. To introduce you to the overall kinase structure here, our compounds bind to the ATP site. That's our ligand is this sort of green blob. And the ATP site is also referred to as the orthosteric site. So I may use those terms interchangeably throughout the talk. Kinases have N and C-terminal lobes, and they're connected by a series of amino acids that we refer to as the hinge. And that kind of just links the two lobes together and wraps right through that ATP or orthosteric site. The other part of kinase structure that I'll refer to today is the G loop, and that's this loop that resides directly over the ATP site. So this is actually a co-crystal structure of compound 2, and if I zoom into the active site, you can see some of the features. We have a hydrogen bond donor acceptor motif along the hinge, as is classical with orthosteric inhibitors of kinases. We see a CH pi stack with our phenyl group over this glycine, and then our basic amine is forming an interaction with an adjacent acidic residue as well as with a residue on the G loop. We additionally see a hydrogen bond with the catalytic glycine there. So as we considered optimization, we recognized that we'd be able to increase lipophilicity to slide up the lipid line. And since 1 unit log D corresponds to 10-fold potency, if we got back up to 1.7, we'd probably be about 20 nanomolar there and in a more reasonable log D space for obtaining desired permeability. So we looked for places to add grease. We wanted to methylate the benzylic amine and add additional steric bulk on our isopropyl group. And we also saw kind of a pocket leading toward the hinge where we thought we could extend from the highlighted position. So the next compound I'm showing is a 3-component change where we've added the steric bulk toward the G loop, alkylated the nitrogen, and extended our isopropyl group with an extra carbon here. So as a composite effect of that 3-component change, compound 3 is about 200-fold more potent, sub-nanomolar Ki, the log D is 1.4, and we do see an increase in lipophilic efficiency. So I haven't shown all the matched pairs here, but we believe a large part of that efficiency increase is the methoxy group. So this drives more potency than the rest of the modifications here. And we believe that's because of optimal positioning in the pocket leading back toward the hinge. So I'll show a co-crystal structure of compound 3 here, and you can see, the additional methyl groups that we added on these vectors, and then, as I alluded to, the positioning of the methoxy group back towards glycine 95. So we see, a solvent front water here, potentially with low resolution in the structure, but also, a kind of close but complementary contact with glycine 95. So this fits with our understanding of the SAR. To expand on the profile of compound 3, of course, we're picomolar in our biochemical assay. As we consider ADME properties, we see low in vitro clearance, high permeability, and high solubility. As we profiled this compound in cell, however, we saw a significant biochemical to cell shift of about 500-fold, which is more than we anticipated given the Km of our enzyme and the ATP concentration in cell. So we're not as potent as we would like in cell at about 250 nanomolar. This phospho-SLP76 assay is one I'll refer to throughout the talk. SLP76 is the downstream substrate of HPK1, so HPK1 phosphorylates it. If we inhibit HPK1, we're inhibiting that phosphorylation. So you'll see SLP76 IC50 throughout. The other assay that you'll see throughout that I wanted to introduce is this PBMC IC50 assay. PBMCs are immune cells, and so they're our target cell of interest, and we're looking to take the brakes, off the immune system, which could lead to proliferation of this cell type. This assay is looking at inhibition of ATP turnover, so it's kind of a cell health assay. We don't want to be doing anything to immune cells that causes them, toxic, death, anti-proliferation, anything like that. We want them to be functioning well not inhibiting them. So this is, as I said, a cell health metric that you'll see throughout. And an unfortunate part of the profile for this compound is that we are inhibiting these PBMC's ability to function just at a fourfold window relative to our on-target activity. So this is a problem that we think indicates poor broad kinase selectivity. So we also ran on this program kinase mini panels that were enriched in kinases with immune cell health function, as well as kinases with high homology to HPK1, meaning they were very, very similar. This heat map is that data for compound 3, and you'll see that we've colored it by window over HPK1 Ki. And there's a lot of red. There's a lot of kinases that we anticipate less than 20-fold window. So we're probably co-inhibiting a lot of kinases with this compound, and that's consistent with this cell health readout. So before I move on, it is really important to underscore that selectivity was important not only for safety, but for efficacy. If we inhibit HPK1 and we turn on the immune system, but then we inhibit a bunch of other kinases that turn it right back off again, we're going to be in trouble in terms of driving our desired therapeutic effect. So selectivity will be a theme of this optimization work. So first focusing on the moderate cellular potency of compound 3. Continuing down the vein of structure-based drug design, we recognize that inner co-crystal structure, the carbon-carbon bond highlighted with the blue arrow, incurred quite a bit of ligand strain. In the bound pose indicated by the cyan, you can see that we have over 4 kcals per mole ligand strain, where the low energy conformation is that where the amine is 90 degrees above or below the plane. We believe that in our co-crystal structures, we pay this high cost for strain because of optimal electrostatic interactions with the adjacent aspartic acid. Because of this, we thought we could improve potency through reduction of ligand strain in the bound conformation. And to do that, we added a pyridyl nitrogen. So if we run the ligand strain assessment around the same carbon-carbon bond, we see that this aza derivative of compound 3 has less than half a kcal per mole strain in that bound conformation. However, we chose not to make this compound. The reason being is that that nitrogen also has an effect on the adjacent methoxy group. This is the preferred conformation, which is a dipole minimized conformation of the lone pairs on each heteroatom. And so this pose is preferred by 2 kcals per mole. So if we made this compound, we'd be rotating this methoxy group away from what we've already determined is a very important pocket for driving potency and efficiency. So we went these direct matched pairs and instead pursued these NN dimethylamines, which would allow us to minimize the ligand strain and also maintain filling of the back pocket as the methyl group needs to go this direction. So this is the same lipid plot that I showed in the introduction, and it shows some really important SAR for this program. What you're seeing in blue are all of the lactams that we had made to date when we initiated those aza derivatives, which are shown in green. What you can see is compound 4 and 5 are matched pairs, and 6 and 7 are matched pairs. And what we observe when we add that nitrogen is an improvement in potency with a reduction in lipophilicity that leads to up to a 0.8 improvement in lipi. So it was really exciting to see that efficiency increase. It was also exciting, as you can kind of tell, most, if not all of our lactams didn't break the 100 nanomolar mark in cell. And compound 4 was actually 80 nanomolar. And while that's not significantly lower than 100, that was a really hard threshold for us to surpass. So we were really excited to see that data come through in cell as hopefully a harbinger of what we could do with this series with future optimization work. So we shifted gears and focused entirely at this point on the aza lactams. So I'm going to bring a multi-component optimization thread to this story, and I'm going to move away from potency for a minute to talk about another liability that came up for us in the course of this optimization, which was high fraction metabolism by CYP2D6. Of course, cytochrome P450s and other enzymes in the liver often metabolize our compounds, clearing them from the system. So why is high fraction metabolism by a specific CYP bad? This is because this specific CYP is polymorphic. It means it has a range of expression differences across the human population. And so there will be people who are poor metabolizers, so the compound sticks around more, you have higher exposure, and extensive metabolizers, people with low exposure. What it ultimately boils down to is variable PK exposure for that compound would be anticipated. And this is problematic because if you don't have a high therapeutic index, you might have people with, sub-efficacious exposures, or on the other end of the spectrum, people with toxicity. And so HPK1, when we worked on it, was a first-in-class mechanism. We didn't know what it was going to do in the clinic necessarily, and you can certainly rationalize that, therapeutic agents that turn on the immune system might have, an autoimmune risk. So we wanted to address this fraction metabolism by 2D6 in order to get the safest compound we could to move into the clinic. So the pharmacophore for CYP2D6 substrates is a basic center that's 5 to 7 angstroms away from the site of oxidation, and it typically includes a flat hydrophobic area. So you can kind of see that in our structures here across the lactam, mesolactam subtypes, which had upwards of 70–80% fraction metabolism by CYP2D6. We did med-ID studies on compound 5, and indeed the methoxy group was the soft spot for CYP2D6, and we hypothesized the N,N-dimethylamine would be the analogous site for our AZA series, although we never did the specific med-ID studies on those compounds. Our computational chemist did some modeling in the CYP for us to take a look at, and you can certainly rationalize that by presence of acidic residues that could interact with our amine, it could orient the soft spot down toward the heme here, which is where we'd undergo oxidation, demethylation, that type of thing. So to mitigate this risk, it was important not only to reduce CYP2D6 metabolism, but do it in a way that didn't reduce other metabolism. If we reduce both in tandem, your fraction or your metabolism or your FM stays the same. So in order to achieve that, we used a LIP-MET-E approach. So what we wanted to do was decrease metabolism by CYP2D6, which is shown on the y-axis, but do it in a way that increased lipophilicity. Since lipophilicity trends with clearance, more lipophilic compounds should be cleared faster, we thought this would ensure that other enzymes would step up and still clear our compound to give us a lower FM. So compound 7 was shown on the previous slide, 80% FM. We shifted to this azetidine, and we did reduce turnover in our recombinant CYP2D6 assay, but the FM stayed the same. We have a very similar log D, and we probably just reduced overall metabolism. So that's an example of what we didn't want to do. Going to the methyl ethyl and gem diethyl, we were able to reduce 2D6 metabolism with increased lipophilicity and achieve appreciable reduction in FM. We also made these really sterically bulky azetidine and pyrrolidine analogs and kind of pushed that to an extreme where with compound 12, we saw barely any turnover by 2D6 and about 0% FM. This was cleared by other enzymes. So this showed an important proof of concept in terms of how we could modulate the soft spot deliberately in conjunction with, tuning the log D to achieve our desired effect. However, the path that proved most fruitful was one we didn't anticipate, one that just came out of routine screening. So compound 7 is an isopropyl triazole. We moved to the N-propyl triazole. We kept the same potency in cell, and we saw a reduction of 2D6 FM. So this was kind of, advantageously appreciated through, like I said, routine screening. So pursuing this N-propyl subseries, we pursued modification of our hinge vector, adding additional lipophilicity there increasing our log D and increasing our potency while maintaining a low FM and 2D6. And compound 15, this cyclic pyrrolidine, this methylpyrrolidine, I should say, gave us our highest cell potency at the time at 34 nanomolar with, again, low metabolism by 2D6. So while we're able to mitigate that metabolic liability and improve our potency, I will comment on the selectivity. So we've seen expanded windows in our cell health assay up to 20-fold, which, as you recall, compound 3 was only 4-fold. So this is an exciting improvement as we've continued our optimization. We do, however, see several kinases that are still co-inhibited with HPK1. So while these compounds are exciting, we thought there was more to do in terms of selectivity. So when I say there's a lot of kinases with high homology to HPK1, this is another way of kind of quantifying that. This is looking at all of the ATP site residues that kind of come into contact with your inhibitor or in close proximity, and just the percentage homology. So there are over a dozen, maybe 2 dozen kinases with greater than 83% homology relative to HPK1. And the coloring is by role, and you'll see that a lot of them have very important roles. So the red ones modulate the immune system, have the potential to obliterate our efficacy if we inhibit them. And the yellow ones, the MSTs, have roles as tumor suppressors. So all very important to avoid, all very similar to HPK1. To emphasize that point, I'm showing a co-crystal structure of one of our inhibitors in green. In HPK1, the protein is green as well. And 2 high homology kinases, MAP4K3 and MST2 in blue and gray. And so the point differences, the amino acid differences that comprise, this 89% homology are shown in stick. Everything else is the same if it's not shown. So you can see phenylalanine to tyrosine, that's just a single heavy atom difference. There is a discrepancy here in the hinge, but it's really pointed away from your inhibitor, and the rest are localized on that G loop. This is an added challenge because the G loop is already a dynamic region. It doesn't mean it means the inhibitor may not be seeing all of these residues. So we did recognize something very unique about HPK1, and that is at this position where our other kinases have branched amino acids, we have a glycine. And in fact, there are 2 conserved glycines on either end of that branching. So HPK1 had a GGG sequence. And as we took a step back and looked across the entire kinome, 400 plus, across 400 plus proteins, only 8 other kinases have a glycine at that residue. They're listed here, and these kinases are not otherwise similar to HPK1. So we hypothesized that HPK1 has a uniquely flexible G loop. And if we extend steric bulk toward that G loop, we may maintain potency against HPK1 and lose potency against our off targets. So the first kind of structural subtype that I'll share is functionalization of our triazole. And so from compound 14, which I shared previously, we simply added a methyl. This methyl maintained potency. And if we look at our functional selectivity readout using our PBMC cell health assay, we expand our window from 24-fold to greater than 41-fold. If we look at our heat map, which I'm not showing on this slide, we do see improved selectivity over specific off targets, including MST3, 4, MAP4K3, and others. So this was really exciting. And compound 16, by virtue of its overall profile, potency, selectivity, metabolic risk, low metabolic risk, this was advanced through tox studies. And so here are exposures for a cyno-TK study. Shown in blue are exposures in male cyno, and shown in red are exposures in female. The solid lines are exposures of the parent, that's compound 16. We did see very high exposures of a metabolite, which I've labeled M1, that's the dotted line. And in fact, based on CAV, we have 8-fold excess of our metabolite relative to our parent. So this could potentially be considered problematic depending on the profile of that metabolite. So prior to the study, we had made a number of metabolites and kind of knew what they looked like. So we had these in hand at the time. They were either synthesized or isolated from CYP incubation. Compound 17 is a des-methyl on the hinge vector. 18 is desmethylation and subsequent oxidation. And then 19 is demethylation at the benzylic nitrogen. So this compound maintained potency, biochemical and cell relative to 16. These other two lost upwards of 10-fold. We didn't even progress them into cell because we thought they were too weak. So the metabolite we saw in cyno that we believed would be relevant to what we would see in human is compound 17. So this is problematic because it's not incurring on-target benefit, and it's actually exaggerating an existing cardiovascular liability that our series has. So basically, this metabolite's just going to cause cardiotoxicity and not do anything we want to the immune system. So this is our cardiovascular liability quantified by an ex vivo rat aorta assay where we're looking at contractility of those aortic muscles. And ultimately, compound 16 was discontinued in progression because of diminished on-target effect and the safety liability that it would have when you consider the metabolites. So this was disappointing, but in parallel to this work, we had been functionalizing our benzylic amine as a way to increase steric bulk toward the G loop, but from the other side of our molecule. So relative to compound 15, which I showed previously, compound 20 and 21 add this primary branched methylamine, and they have one of two triazoles, like an alkylation, in this case an ethyl, or this rigidified cyclic triazole. These compounds are comparable potency to 15, if not improved, and similar to what the methyl group on the triazole did, we're improving our cell health using this PBMC assay upwards of 40-fold. So compound 21 is the clinical candidate, and I'll show a little bit more on its profile here, starting with its co-crystal structure. This is a really beautiful high-resolution 1.5 angstrom co-crystal structure, and you can see that amine forming a directed interaction with the asp in what should be a low ligand strain conformation. That branched ethyl resides right below that unique GGG sequence in HPK1. I mentioned kind of the cell health readout on selectivity, and shown here is the same sort of heat map that I showed early in the talk, where we're coloring by selectivity window. Most of these kinases are greater than 100-fold window relative to HPK1 Ki. There's a few in the 20 to 50-fold range. This compound is remarkably selective, especially considering, the starting point, how much red we had before. So compound 21 is much more complicated than compound 16. 16 was discontinued due to active metabolite safety implications. This table kind of shows you what the synthetic complexity buys you. It's about 5-fold potency in cell. And if you look at selectivity at over a 395 kinase panel, the percentages of kinases inhibited less than 30 and less than 100-fold window, you go from 2 and 6% to less than 1% and 2%. So compound 21 is more selective. Further, the increased on-target activity buys you a much expanded window in regards to that cardiovascular liability. So compound 21, as I mentioned, was advanced to phase 1 clinical trials in recent years. This is a bit of its expanded PK profile. So we have low in vitro clearance, high permeability, and high kinetic solubility at pH 7.4. We predicted from our hepatic clearance, blood clearance of 5 mils per minute per kilogram with a 12-hour effective half-life and moderate to high absorption and bioavailability. So the only sort of efficacy I'm going to share today, apart from the phosphoslip76 IC50, is this really elegant tumor co-culture model. So this co-culture takes A375 melanoma cells and weakly reactive T cells, and we add our compound and anti-PD-1 to observe what sort of control we can obtain in this system. So the A375 cells grow on their own. That's indicated by the open circles. If you add the T cells, they're weakly reactive. They control the system a little bit, but not a lot. So this is exactly the weak antigenic stimulation where HPK1 should play a role. When we add compound 21, we exert control as indicated by the red triangles. We have an anti-PD-1 agent. Anti-PD-1 is effective in the clinic, and there's many approved large molecule of this subtype. So we were excited to see about comparable control relative to saslelimab. When you combine the two, you get even more extreme control. So this was really exciting to us. It's a human-derived system and provides the exact proof of concept that we thought these molecules could function in. So kind of to summarize here, we started with a series of AMID inhibitors and moved to our lactam and azolactam series with increasing very specific steric bulk to drive the selectivity profile we wanted. We started with this early lead, about a 250 nanomolar in cell, and we're able to move down to 17 nanomolar. We moved from entirely unselective, activating and inhibiting immune cells at the same level pretty much, to something with almost a 50-fold window. And that's ultimately how we identified our clinical candidate. So with that, I need to thank and acknowledge a very large project team that did a ton of work over multiple years to identify this compound. We have a ton of chemists across different areas that contributed, and of course, all of our partner lines, PharmSci, DSRD, PDM, and our biology, biochemistry, biophysics colleagues as well. So with that, I'd like to thank you all for your time, and I'm happy to take any questions.
Awesome. Thank you so much, Rebecca. That was a very nice and clear story. So really very much appreciated. We do have questions in the audience already. So there's one question about the target HPK and the kind of modality it can be addressed with. So Gagan says that beyond the kinase function, HPK probably has other kinase-independent regulatory roles in several innate immunity cells, so dendritic or natural killer cells, that might not be addressed by a kinase inhibitor. So he's wondering what your take on a potential PROTAC, so a degrader of HPK, would be.
Yeah, I mean, there's been a lot of activity in the field, both small molecule inhibition as well as PROTACs. And I feel like that kind of conceptual question has been one that the community is interested in across pretty much any target where you can identify multiple roles. I think it's one that maybe will hopefully emerge from the clinic in recent years, or in coming years, I should say. Anytime there's multiple roles, yeah, there's potential for enhanced efficacy. There's also potential for enhanced safety risk. So I think, that's something we'll have to determine through experimentation, I think.
Yeah, sounds good. Brandon.
Hey, Rebecca, great talk. I thought the 2D6 story was great. Those metabolomics type studies are always like some of the ones you remember. And I'm curious whether looking at your candidate, if there was a structure-based hypothesis. I think the 2D6 pharmacophore is an amine stacked between aspartate and a phenylalanine. And if the landing at the primary amine in the end was actually critical then as you move forward with the triazole structure and so on. Yeah, so I think you have a in the back there a little bit.
Yeah, modeling in SIPs is tricky because they're very dynamic.
Right.
So this was kind of like a feel good thing, but it did fit with the pharmacophore and what we were seeing. We did some SAR here. We did see SAR. I don't think consistently you could take off the methyl and get out of this space. Each compound kind of had its own fingerprint because of what other CYPs were metabolizing it as well. So it's not a surefire fix, but yeah, it could have had a role in the end, potentially.
Okay, sure. Thanks so much.
Yeah, I guess our next question is, did you check out macrocyclic versions of compound 21? So I think the audience is thinking about connecting that ethyl group and looping it back to the triazole there.
Yeah, I'm going to go to a co-crystal structure. It's easier for people
Yeah,
to see, perhaps. Yeah, so some sort of connectivity here. We
yeah.
talked about it as a team, but we didn't actually ever go that route. I think macrocycles can, in times, give you great benefit. We did have low ligand strain in the bound conformation here. So we were already at a pretty solid starting point in terms of reinforcing this conformation. But in short, we didn't pursue them.
Yeah, makes total sense. Yeah, sounds good. Next question is from Brian de la Barre. He says, terrific talk, everything very well explained. With the noted flexibility of the HPK G loop and potentially other regions, were FEP methods useful or were they not applied at all?
I'll have to stretch my memory back. I don't think we, yeah, we did it, I'll just say we used docking, we used scoring, we used MD, we used all sorts of methods. I'd have to kind of go back to our comp chem and dig into that a little bit more. But we did have significant comp chem, impact, I'd say, on this program.
Yeah, sounds good. Brandon, did you have another question?
No, I was just going to comment that I think as an ex-Schrodinger employee, maybe dynamic regions are not always the best suited to FEP methods. So
Hmm.
um, because you're not sampling long enough to see a protein movement, and your input depends your output or determines your output rather.
Yeah, thank you.
Yeah. Next question is from Quinn Bumpers. So I think you very nicely explained that cardiac liability with the aortic tissue. And Quinn is wondering, did you profile HERC through lead optimization and did it correlate with the red aorta IC50 or the steric substitution?
Yeah, I mean, that's a great question. So we routinely screened in HERG and, calcium and sodium channels throughout. The right aorta was just like a later tier assay. So we did see mixed ion channel effect throughout, and the predominant impact we thought was more like blood pressure driven, potentially based on disproportionate calcium effect. And that's why we use the rat aorta, which I think reflects that a little bit better. That's just why I benchmarked against that. So all of those ion channels were part of the optimization. I just didn't kind of go into that level of detail. But you're right. Yeah, we used it and it was part of the story.
Yeah, super. Makes sense. Yeah, maybe in the interest of time, maybe one more question, and you have it here on the slide. Actually, Govi is wondering about this. What is the selectivity you observe for the HPK subfamily members, if there was selectivity, and that's in the panel right here? And what was driving the selectivity given the high homology you've talked about? Is it really the G loop flexibility, and that those other closely related kinases do not have that G loop flexibility?
Yeah, so HPK1 is otherwise known as MAP4K1. So MAP4K2, 3, 4, 5. That's why that specific subset is represented in this panel. They're the most highly, homologous, or some of them. So we see MAP4K2 is greater than 100-fold. 3 and 5 were, 20 to 50, and then 4K4 was 50 to 100. So we were able to move those up appreciably. This is based on single point data. We did subsequently get IC50s, biochemical IC50s, and largely think that, we're obtaining those levels quoted here. So they are some of the harder ones to obtain selectivity over, and I do think the G loop was of critical importance for that. We had other series, and we were looking at this G-loop hypothesis there, and it wasn't as effective in those series. And I think sometimes it's just the vectors you have from specific atoms that do better or worse. So it's maybe a long way of saying series specific effect, but I think we were uniquely able to move the needle with this hypothesis in this series.
Nice. Nice. Well, I know we're a little bit over time, and maybe I find this question quite interesting. So maybe we'll do this as the last question. Sorry, Rebecca. So it's from Huan Yu Shi. He said, or they said, I noticed that the P-SLAP76 IC50 values were measured using Jurkat cells, while the cellular toxicities were measured using PBMCs. Why is using these two data? Why can you use two different cell types to calculate the cellular window value?
Yeah, we did. We used Jurkat for routine screening. However, we did look at, phospho-SLIP modulation and other cell types and such. So that was just part of later tier profiling, and we don't necessarily have that for every single compound. So we did believe that the Jurkat was, predictive of what we'd see in terms of phospho-SLIP modulation and other cell types. But yeah, just for ease of presentation and completeness of data, this is what we've included here, I'd say.
Awesome. Awesome. Well, thank you so much again, Rebecca. That was such a great talk, super clear. I think I learned a lot of things about HPK here. So we want to thank you again for a fantastic talk, sticking around to answer the audience questions as well. And we want to thank, of course, our audience for the engagement and all the questions. And very excited.
Thank you so much.
And with that, we'll close out here. So we'll be back next week. We'll have Peter Tumino from Nimbus here, and he'll be talking about designing selective SIK2 inhibitors for dual anti-inflammatory and pro-repair effects in inflammatory and autoimmune diseases. So very excited for that. And we'll also release a nice update to the structure search, which will be called molecule search next week. So stay tuned for that as well. And with that, I wish everybody a wonderful rest of their week. And talk to you all later. Bye now.